Hi there, thank you for coming. Today I'm going to be talking about some work that my colleagues and I have been doing to better understand how thermal performance curves change shape in response to other abiotic factors, in particular the oxygen environment. So to kick things off, I'd like to briefly describe the thermal performance curve. This is a univariate function that describes how some measure of performance varies with temperature. They typically are left skewed and hump-shaped and characterized by a handful of parameters, including the optimum temperature where performance is maximized, and two critical thermal limits, a minimum and a maximum. Now, this basic paradigm has largely guided thermophysiologic research in ectotherms for about 50 years now. But these curves, while very useful, do have a limitation, and that's that these are inherently descriptive with little underlying mechanism. And because they are descriptive, it's very hard to predict how the shape of this curve might change across contexts. And we know that these curves can change shape, and contexts that can do it include length of exposure, the trait that's being measured, pathology, prior exposures, all sorts of things, and we're not very good at predicting them. So what we opted to do was to try to tackle just part of this, and particularly wanted to know how thermal performance curves varied across the oxygen environment and whether we could mechanistically predict this change in shape. And to do this, we developed a somewhat new hypothesis called hierarchical mechanisms of thermal limitation. This largely combines ideas from oxygen and capacity limited thermal tolerance, as well as the marginal stability hypothesis. And to illustrate this framework, I'm going to use this figure. The y-axis here is metabolic rate, and the x-axis is temperature, body temperature namely, and we've got two lines. The blue line is resting metabolic rate. This is the amount of energy, metabolism, that the animal is using just to maintain basic functions while it's sitting at rest. For this line, the data come from pythons at rest, and you can see there's this exponential effect of temperature that's going to be driven by basic thermal kinetics. We then have the red line, which is the maximum metabolic rate. This is the absolute max that the animal can do aerobic work at each of these temperatures. There, you can see that it goes up exponentially and then plateaus. And it plateaus at that dotted line, which would be the maximum capacity for the organism to acquire and circulate oxygen to its tissues. The distance then between the two lines is the aerobic scope for activity. This is the full breadth of metabolic upregulation that is possible for the organism to do aerobic work and maintain fitness-relevant functions that require aerobic work, things like digestion and reproduction. Even foraging, most movement is going to be aerobically powered. In this case, the data points are from published data for pythons, either at rest, or the red line data come from pythons digesting a very large meal. And you'll notice the red and the blue lines do meet at the right of the plot. This is where the aerobic scope drops to 0. They have no potential for aerobic work. They're using all of their aerobic potential just to be at rest. This we call the aerobic T-crit. It's the temperature at which aerobic scope falls to 0, and it could set the critical thermal maximum. However, in this case, the critical thermal maximum is shown by that vertical black line. And we think it's set by subcellular mechanisms. This is proteins or cell membranes becoming too flexible to properly function, and the organism loses function at those temperatures. Now, this becomes really interesting when we consider what happens when you reduce aerobic capacity. That's going to lower the dotted line, which brings the red line down. Aerobic T-crit shifts to the left, and aerobic scope declines, and the temperature where aerobic scope is maximized has shifted left. However, you'll notice that aerobic T-crit has not traded positions with subcellular T-crit, so we don't expect any change in the mechanism driving CT max. We can then look at this in thermal performance curve form this way. So here's the thermal performance curve at normoxia. When we expose the animal to hypoxia, we expect the whole curve to move down and a little bit to the left as maximum performance becomes limited and the optimal temperature shifts to the left. CT max in this case has not been affected because aerobic T-crit is still well to the right of subcellular T-crit. However, it is possible to cause aerobic T-crit to underlie CT max under this framework. But in this case, it would cause extreme hypoxia exposure that brings that dotted line, maximum metabolic capacity, way down. So now the red and the blue lines are crossing at temperatures below the subcellular T-crit. Now aerobic T-crit has shifted to the left. That is now going to drive CT max. And you can see the aerobic scope for activity has gotten much, much smaller. If we look at this effect on the thermal performance curve, we then predict the optimum temperature and peak performance to be dramatically affected with this massive leftward shift and downward shift, causing the whole performance curve to become more symmetrical. CT max has been affected, but quite minimally compared to the strong effect in extreme hypoxia on the other aspects of the shape of the thermal performance curve. So we wanted to test these new predictions of the hierarchical mechanisms of thermal limitation hypothesis, namely that TPC shape will change with reduced oxygen capacity even if CT max does not change. We're going to expect peak performance to be reduced, the optimum temperature to be reduced, and the curve to be more symmetrical when animals are exposed to hypoxia. We really expect CT max to be quite robust and possibly not be affected by hypoxia at all. And we tested these predictions using the common wall lizard, which is a widespread lizard native to Europe, and they are actually migrating uphill in response to climate change. And we really want to know, can they completely invade up the mountain and possibly threaten some endangered high elevation endemics? We collected the animals all at low elevation. We then measured their performance for 2 different traits at that same low elevation of capture, or at a research station on top of a mountain that's about 600 meters above the natural range limit for the species in the area. So this is an area they don't currently live in, but they potentially could invade. And we measured performance across 7 temperatures at each of these sites for 2 performance measures. We started with sprint speed. We selected this because it's a very common measure of whole organism performance in reptiles, especially lizards. However, it's 80 to 90% powered by anaerobic metabolism, meaning that this measure of whole organism performance could be particularly robust against variation in the oxygen environment, but it also might give us a false signal of how robust the whole organism actually is. So we also wanted to examine aerobic performance. And for that, we used aerobic scope, now measured in our lizards, not by gut loading them, which doesn't really work to get at maximum metabolic rate in lizards, but instead we perturbed the lizards in the respiration chambers with a small remote controlled car that got them to sort of run around in the respiration chamber. This is a somewhat novel method. We never saw injury on any of the animals. It seemed to work quite well. We got nice level traces for maximum metabolic rate. So we used that to get MMR. We also measured RMR for an animal at rest and used that to estimate aerobic scope for activity. We then developed performance curves by fitting the data to the beta function. This function is one of the many used to describe thermal performance curves and ectotherms, but a recent meta-analysis showed that it really does the best job for lizards and other reptiles. It best fits the data, and it has parameters that are readily biologically interpretable. These include WF, which is the peak performance, T-opt, the optimum temperature, or the temperature where peak performance occurs, and then the CT max, the temperature where performance declines to 0. Now we actually only fit the model to estimate WF and T-opt. We did not estimate CT max through model fitting because a recent study using the exact same population of animals that was carried out by Brooke Bodensteiner and colleagues found that CT max is unaffected by elevation in our lizards. So that supports one of the predictions of hierarchical mechanisms of thermal limitation and allowed us to use a fixed value of 41.3 estimated from that study to be the CT max when we were fitting this beta function. The primary results from this analysis are shown here. We have temperature on the x-axis, performance on the y-axis, sprint speed is shown on the left, aerobic scope is shown on the right. The purple line and shaded region is the thermal performance curve for high elevation lizards and their 95% confidence interval, whereas the tan is performance for lizards measured at low elevation. And what we found was that at low elevation, the 2 curves are quite similar, sprint speed and aerobic scope. However, at high elevation, they differ quite dramatically, with sprint speed being unaffected at high elevation, while aerobic scope, the thermal performance curve has shifted exactly as we predict from hierarchical mechanisms of thermal limitation, with the peak performance dropping markedly and the optimum temperature for performance shifting to the left by approximately 2 degrees C. So the next thing we really wanted to do was be able to quantify the effect of the oxygen environment on those thermal performance curves. Now in this case, we can only estimate them at a population level, but once we quantify this sensitivity, we could use those quantified sensitivities to compare either among individuals or species in the future to better understand this physiological interaction. So to do this, we updated that beta function for the thermal performance curve so that we could model temperature oxygen performance surfaces, which we call TOPS, and an example of which is shown spinning at the bottom right. We did this by substituting into the beta equation these functions shown on the bottom here for either WF or T-opt in the beta equation. This added 3 new parameters, or 2 new parameters and 1 state variable. The state variable is the fractional oxygen environment, which is the ratio of oxygen availability or capacity during your test divided by a reference. And then we have 2 oxygen sensitivity coefficients. Gamma describes the sensitivity of peak performance to the oxygen environment, and tau represents the sensitivity of the optimum temperature of the thermal performance curve to the oxygen environment. So we fit these for both sprint performance and aerobic scope, which resulted in the following temperature oxygen performance surfaces, and their parameters, the estimated parameters, are shown on the bottom. We can see that for aerobic scope, there's this steep drop caused by the oxygen environment, where we do not see that at all for sprint speed, indicating that definite robustness of sprint speed to the oxygen environment. This is directly quantified with the gamma and tau parameters. We can see that gamma is approximately 8-fold higher than for aerobic scope than it is for sprint speed, and tau is, almost infinite-fold higher because it is a value of approximately 0.2 compared to a value of approximately 0. So we see much greater sensitivity to the oxygen environment for aerobic scope than we do for sprint speed. And this observation matches our predictions from the hierarchical mechanisms of thermal limitation hypothesis, especially for that aerobic performance measure. We saw TPC shape change exactly as predicted, with peak performance being reduced, T-op shifting to the left, and the curve becoming more symmetrical, while CT max appeared quite robust and was not changing in response to our elevation changes. Now, importantly, the distinction we see between sprint performance and aerobic scope can help us understand some problems and predictions for how organisms are going to respond to climate change. Because if we just look at sprint speed's thermal performance curve, we would expect Podarcis moralis to be able to readily persist and invade our high elevation site. However, if we look at aerobic scope, we would expect them to not be able to do this very easily. They're going to have a very different thermal performance curve at that high elevation with a rather flat amount of aerobic scope across temperatures. And it turns out when we actually bring animals to this high elevation and we allow them to be up there for a while, we find that they lose mass even when they're fed ad libitum over about a 6-week period. We think that's because they have this very limited aerobic scope. They cannot do aerobic things like digestion. So they cannot persist at this high elevation, even though their sprint running is unaffected, which I think really illustrates how important your selection of trait is as a fitness proxy. And I think if we're trying to look at sort of the effects of chronic changes in the environment, we might really want to have an aerobic trait rather than an anaerobic trait be our proxy for fitness. Moving forward, we would like to expand on this TOPS framework. In particular, we would like to estimate TOPS for individuals rather than populations, as we did here. Once we can estimate it at the individual level, we should be able to compare gamma and tau statistically to understand variation in this physiology. We'd also like to be able to compare species and populations and test for plasticity. And then conceptually, this framework could be extended to include other abiotic factors, developing hypervolumes rather than just performance surfaces. We've begun some of this work, especially for estimating individual level TOPS and comparing species. If you're interested in that, I encourage you to check out the presentation by my former student, Dalton Leibold. It's number 40-2. With that, I'd like to thank the many people that helped out with this work, including past and present students in my laboratory at Fresno State, as well as the great team of researchers we were able to bring together to do this work, multiple funders, and thank you all for being here. Please leave any questions you have in the chat, and I will do my best to get to them. Thank you for your time.